Chapter 9, Topic 9.3, Chemical Agents and Microbial Control. In this topic, the objectives are: name the desired characteristics of chemical control agents, discuss chlorine and iodine and their uses, list the advantages and disadvantages of phenolic compounds, explain the mode of action of chlorhexidine, explain the applications of hydrogen peroxide agents, identify some heavy metal control agents, discuss disadvantages of aldehyde agents, and identify applications of ethylene oxide sterilization.
Under the heading of chemical methods, we're going to be discussing how one evaluates a disinfectant. Then we're going to discuss the active ingredients that are found in disinfectants, chemical sterilants, and the like, including phenol and phenolics, halogens, alcohols, heavy metals and their compounds, surface active agents, chemical food preservatives, aldehydes, chemical sterilization, and peroxygens and other forms of oxygen. First, let's start with a question. You're in Walmart or some other store in the area, and you're confronted with this. All sorts of cleaners, sanitizers, disinfectants, you name it. How do you pick? Now, as a consumer, most of us pick first on cost, and second on how well it cleans, things that we can see, how well it removes dirt and such, and thirdly, and I hate to say this, by whether it stinks or not, how nicely it smells. What we really should be picking it on is how effectively it controls microbial growth, but you have no way of telling that. You can't tell by looking at a surface whether you sufficiently reduce the number of live microbes or not. So we have testing labs that go through and do this kind of thing. As we go through this portion of the chapter, you're going to be learning about how effective different active ingredients are, and this will enable you to be better able to pick the proper chemical control method for what you're trying to do. Now let's talk about how we evaluate a disinfectant. There are three main tests that are currently common in the United States: the in-use dilution test, diffusion tests, and in-use testing. First off, let's talk about use dilution tests. You're going to be doing one of these in labs, and here we have a picture of a bunch of test tubes. They have decreasing amounts of the disinfectant in the test tubes. Okay. Then what you do is you add the same amount, the same number of microbes, and we generally pick, you can think of it as a rogues gallery of microbes, the ones that cause the most problems clinically, and also those that are hardest to kill. And we look and we see where the tubes become turbid. Okay, right here you can see it's turbid, right here you can see it's not. Right here is the minimum concentration of the disinfectant that has inhibited growth of the microbe. Now at this point we don't know if the active ingredient is killing the microbe or stopping it from growing, and depending upon what you do, doesn't really matter. Okay, so we call this the MIC, the minimum inhibitory concentration. Okay, and this is use dilution. This tells us the smallest dilution, or rather, excuse me, the other way around, the highest dilution that we can use and still inhibit the microbe. For this next one, the diffusion test, we generally use this for antibiotics, but you can use it for surface disinfectants. And since there's a good picture in the book, we're going to use this as an example. Here we put down on agar a lawn of bacteria. Then at the same time we drop a disc that has been soaked in the active ingredient. Then after incubation we see where the organism has been inhibited in its growth. It's kind of like the use dilution test that we looked at previously, okay, where we've got inhibition of growth after incubation in tubes, but in this situation we've got it on a solid surface. And once again, we've got the MIC. Right here at the boundary is the minimum inhibitory concentration, because the active ingredient has diffused out of the disc into the agar, and we see where the minimum inhibitory concentration is. Now the only problem is with the disc diffusion test is that we don't know what that MIC is. You have to go in and take a sample of the agar and figure out how much of the active ingredient is in there. Now with antibiotics, they've done that test and they've figured out what the diameter is for where the MIC is. Okay, and we'll talk about that more when we do the disc diffusion test for antibiotics in lab. But before we move on, there's one thing that I do want to point out. Each of these discs has the same amount of active ingredient on it, but we're looking at 3 different microbes. So you can see here that our gram-negative organism was not particularly sensitive to our phenol here, our hexachlorophene, nor the O-phenylphenol. This gram-negative wasn't sensitive to this one, but it's starting to have some effect here. Over here you can see that our gram-positive is highly sensitive. So if you're looking at the same amount of active ingredient, same agar, same amount of diffusion, and you're looking at 3 different organisms, you can see how sensitive those organisms are to that particular active ingredient. This becomes important later at the end of this chapter when we talk about the resistance of various microbes. It's these kinds of tests that allow us to determine this. And when I say these kinds of tests, I'm including the use dilution tests in it. Now for in-use testing. That's where you take a model area, model operatory. I happen to have a dental operatory here because that's what I worked in for many, many years, testing disinfectants. And that's where you take a model operatory, in this case a dental operatory. It could be a surgical operatory in a hospital. It could be an model exam room that a health clinic would have, whatever the situation is, and you test how well the disinfectant does in a real, unquote, situation. So what you would do is you would take and you would swab the area before you apply the disinfectant, try to pull out how many organisms that you can. Then you would go through the disinfectant, wipe everything down, follow the directions, and then re-swab to see what the reduction in the number of microbes is. Sometimes you have, you would actually put a person in, do a dental procedure, and so you would swab the area after you've had saliva spraying all over the place from the dental drill, and then you would swab before, disinfect, swab after. Now, in-use testing is becoming more and more popular because in the lab everything's perfect. In real life, people don't follow the directions, and they're in a hurry. In active dental offices, they generally have 3 minutes in between patients, and so they're not going to let that disinfectant sit for the recommended 10 minutes. Nobody lets the disinfectant sit on the counters for the recommended 10 minutes. I mean, it's kind of silly to require that, but so this is becoming more popular, and we have a tendency to use it with the use dilution tests, the perfect in-lab testing, so where we know exactly how much of the organism we have, we know what the concentration of the active ingredient is, combining it with real-world testing. Now on to the different active ingredients. Let's start with phenolics. Okay, I have a bottle of antibacterial hand soap because the active ingredient in that is a phenol. Here's the chemical formula of a phenol. Back at the beginning of learning to use disinfectants, Lister of Listerine fame would use carbolic acid or phenol as a surgical dressing, and also he would spray the wounds before and after surgery of the patients that he was working on, and they had a reduction of sepsis in the patient, of infection in the patient. Well, phenol in highly concentrated form, such as carbolic acid, causes cancer. So we don't use it in its highly concentrated form, and in fact we generally use it in a biphenol form because it's far less toxic. You could drink this stuff in most of its concentration that we use and it's not going to harm you, but it harms microbes. Okay? And because it was phenols and their derivatives were one of the first to be used as a surface disinfectant and as an antiseptic, that we still have a tendency to go back to that as the gold standard. Okay, so triclosan, you want to remember triclosan. Triclosan, if you look at your antibacterial soaps, you'll see triclosan listed. You'll see it listed on garbage can liners that advertise reducing smell. Well, if you decrease microbial growth, you decrease the smell. We put triclosan in all sorts of stuff, everything from baby diapers to cutting boards to shower curtains, you name it. Now the other phenol, or phenolic, that I want you to know is Hibiclens. Its active ingredient is chlorhexidine. It's a, I'm not even going to make you learn how to say that, but you've got two phenol derivatives. We've replaced the OH group with a chlorine, and we've got this nice amine containing molecule that hooks it together. Now this means that it's active for longer. It takes the body and the environment longer to break it down, and you'll find chlorhexidine in a lot of antiseptic uses, kind of like triclosan. But with this one, Hibiclens is used as a antiseptic surgical scrub that various clinicians use before they don their gloves. And then also you will find chlorhexidine in oral mouth rinses that patients will use before oral surgery. The microbes in your mouth are your friends until there's going to be cutting to be done, and then you really don't want them in your bloodstream. So you want to decrease the numbers so that your immune system can handle them so that you don't have other infections. Now before we leave phenolics, let's talk about how they act to control microbial growth. They get in and they disrupt cell membranes. Okay, you can see how they would do it. This part is, you got it, hydrophobic, inserts into the cell membrane. You've got your other groups on there that act to pop those cell membranes. So I want you to remember the two examples of phenol or phenolics, and I want you to remember how it acts against cell membranes. On to the halogens. Now remember halogens from the infamous chapter 2. Halogens are the atoms that need just one more electron to have a full valence electron shell. So what do? They go around stealing electrons. Okay, and that wreaks havoc on microbes. Now the most common halogen is chlorine. Now bleach is sodium hypochlorite, and that's the one that you're most familiar with. Not only does it make your clothes whiter, but it kills microbes. We also use chlorine gas in treating city water and swimming pool water, such things. And what happens is that chlorine gets there and it impairs the activity of the enzymes. So basically it shuts down the enzymes function. Bromine, which is one down on the periodic table, it's a little bigger molecule, doesn't evaporate as fast. We have a tendency to use bromine for treating hot tub water. It's hotter, the chlorine would evaporate too fast, so we put bromine in, doesn't evaporate quite as fast. Iodine is also a halogen. It's one down from bromine. Now because it's even bigger, its mode of action is a little bit different. It's still stealing electrons, but what it does is it forms complexes, at least so far as we know. It forms complexes with amino acids, and that inhibits protein synthesis. They also bind to unsaturated fats. Remember, an unsaturated fatty acid is one that doesn't have all of the bonds of a carbon taken up with a hydrogen, so it has some double bonds. So chlorine comes in, steals the electron from the double bond, breaks that double bond basically, straightens out the fatty acid. It's no longer the right shape. So halogens are highly effective in small quantities. Well, at least with bleaches, with your chlorines, there's a problem. And you probably already guessed it. If you're using that for a surface disinfectant, it's going to bleach everything. It's going to bleach your countertops, it's going to bleach your clothing, it's going to bleach your carpets if you spill it, and it doesn't smell very good. So we have a tendency to dilute it out And we also have a tendency to use other surface disinfectants that aren't quite so caustic because, well, by caustic, one thing I forgot to mention, it also steals electrons from your metals. In other words, it corrodes your metals. So we have a tendency to use halogen sparingly or to bind it to other things that kind of reel in its electron stealing capabilities. On to the alcohols. Now you're familiar with isopropyl alcohol, rubbing alcohol, in its use as an antiseptic, in its use on human skin. Okay, it's highly effective. Not so good as a surface disinfectant. It's really quite good as an antiseptic, not so good as a surface disinfectant. If you take ethanol and use it in high quantities, such as in Lysol disinfectant spray, then that's much better as a surface disinfectant. Alcohols act by disrupting cell membranes. Okay? They also dehydrate cells. So those two things, pulling water out of cells, busting up the cell membrane, is how they kill. Now, one of the problems with alcohol is that it evaporates very quickly. Okay? So with rubbing alcohol, that's not a problem. You're putting it on the skin to reduce the number of microbes, either after you've had a break in the skin or when you're going to put a break in the skin, like for hypodermic injection, etc. You do want it to evaporate quickly. When you're using a surface disinfectant, you don't want it to evaporate quickly. So part of the thing is that we put it in this aerosol can with these bottles of isopropyl. Every time you open it up, you get a little bit of alcohol evaporating, and so your concentration of alcohol to water goes down. So you might start out with 70%, but by the time you're done, you're more like 30%. So we put them in these pressurized canisters so that air doesn't get in as you force it out. And also we put in other chemicals that help the alcohol to lay down so it doesn't beat up on your countertops. And we'll talk more about those surface active ingredients later on.
Well, I've gotten rather long-winded talking about stuff that I used to do for a living. Anyway, I'm going to stop here, and then we will pick up the rest of this topic in part 2.